Hello everyone, welcome to Gyaan. Today, we're diving into aging. But we're not just talking about wrinkles and gray hair. We're going deep into what happens inside your cells as you age, why some cells stop dividing, how this connects to cancer, and how your body has a built-in self-destruct system called apoptosis. This topic is directly from Gray's Anatomy, and I've also researched the key scientific papers cited in it, so you're getting the full picture today. Let's get started. So, what exactly is aging? In simple terms, aging is a gradual decline in the function of your cells and tissues over time. Think of it like a machine that slowly wears out. The parts don't work as efficiently as they once did. Now, here's an important point. Aging does not always mean a shorter life. But it does increase your risk of disease. Scientists have identified specific hallmarks of aging. These are the key cellular changes that drive the aging process. They include genomic instability. Your DNA gets damaged more often. Telomere shortening. The protective caps on your chromosomes get shorter with each cell division. Mitochondrial dysfunction. Your cells' powerhouses become less efficient. And cellular senescence, which we're going to discuss in detail in just a moment. This framework was beautifully reviewed by Cubbon and Miss Steele in 2017, published in the prestigious journal Nature Reviews Molecular Cell Biology. They focused on how premature aging diseases, like Hutchinson-Gilford progeria syndrome, share the same cellular mechanisms as normal aging. Alright, let's talk about cellular senescence. This is one of the most important concepts in this entire topic. In simple terms, cellular senescence means a cell has permanently stopped dividing. I want you to imagine a photocopier that has permanently jammed. It can't make new copies, but it's still plugged in, still running, and it might even be causing problems for the other machines around it. That's exactly what a senescent cell is. Now, there's an important difference between a quiescent cell and a senescent cell. A quiescent cell is like someone on vacation. They're temporarily paused, but you can call them back to work. Give them the right signal, and they'll start dividing again. A senescent cell, on the other hand, is permanently retired. No matter what signals you send, it will not divide again. It's stuck in this arrested state forever. And what causes senescence? Three main things. DNA damage at the telomeres, when your chromosomal caps get too short. Decreased mitogenic signaling. The cell is no longer getting the right growth signals. Activation of tumor suppressors combined with genomic damage. This was covered in detail by Chandler and Peters in 2013, who explored the relationship between senescence and the cell cycle. Now here's where it gets really clinically important, because senescent cells accumulate with age, and they contribute to some of the most devastating diseases we know. Look at this list. Alzheimer's and Parkinson's disease, neurodegenerative diseases where brain cells progressively die. Atherosclerosis leading to stroke, plaque builds up in arteries, cutting off blood supply to the brain. Osteoporosis, bones become weak and fragile. Macular degeneration, loss of central vision in the elderly. Cardiopulmonary failure, the heart and lungs stop working efficiently. Renal failure, kidney function deteriorates. All of these conditions are significantly driven by the accumulation of senescent cells in old age. This was documented by Tuttle and colleagues in 2021. So when we talk about aging diseases, we're essentially talking about the consequences of cellular senescence piling up in our bodies over decades. Now here's the really fascinating, and slightly scary part. Senescent cells don't just sit there quietly. They actively secrete signals into their surroundings. This is called the SASP, the Senescence-Associated Secretory phenotype. Think of it like a smoke alarm going off and never stopping. The alarm is supposed to alert the body that something is wrong. But in old age, these alarms never switch off, causing chronic, low-grade inflammation throughout the body. SASP involves three major types of secreted molecules. 1. Pro-inflammatory cytokines. These trigger inflammation. Short term, this is useful for healing. Long term, it causes damage. 2. Growth factors. These stimulate stem cell renewal and tissue repair. Again, useful short term, but if uncontrolled, they can stimulate abnormal cell growth. 3. Proteases. Enzymes that remodel the tissue around the cell. These can break down healthy tissue if overactive. And here's the clinical bombshell. SASP may also make cancer cells resistant to chemotherapy. This is an active area of research and has major implications for how we treat cancer in elderly patients. This was described in detail by Dr. Judith Campisi in her landmark 2013 review in Annual Review of Physiology, one of the most cited papers in aging biology. So scientists want to study senescent cells, but how do you actually identify them under a microscope? There are two main markers. Marker 1. Beta-galactosidase, or SA-beta-gal. This is a lysosomal enzyme. Lysosomes are the cell's recycling bins. In senescent cells, this enzyme becomes overexpressed, meaning there's a lot more of it than normal. Scientists use a special stain that turns this enzyme blue, so senescent cells literally show up as blue under the microscope. Marker 2, P16INK4A. This is a tumor suppressor protein. Its job is to apply the brakes to the cell cycle. In senescent cells, P16INK4A is highly expressed and it promotes the formation of SHF, Senescence-Associated heterochromatin foci. Basically, the DNA gets packed away tightly so the cell can never divide again. High levels of P16INK4A equals a senescent cell. These two markers are used in research labs and even in clinical studies to detect and map senescent cells in human tissues. Now we come to one of the most paradoxical relationships in biology: senescence and cancer. Because senescence is both a friend and an enemy when it comes to cancer. The good side. Senescence suppresses cancer by permanently stopping damaged cells from dividing. Remember, cancer needs cell division to grow and spread. Senescence applies the permanent brakes. When a cell experiences an oncogene being switched on abnormally, it can undergo OIS, oncogene-induced senescence, essentially putting itself into permanent arrest to prevent becoming cancerous. It's the body's safety net. The bad side. But here's the problem. Senescent cells can actually promote cancer in neighboring cells. How? Through SSB. The inflammatory signals released by senescent cells can stimulate nearby premalignant cells to proliferate and progress to full malignancy. And cancer cells are smart. They develop mutations in the p53 and p16 retinoblastoma pathways specifically to escape senescence. These mutations allow them to bypass the normal stop signals and keep dividing. So senescence is a tumor suppressor when it works, but when cancer cells evolve to evade it, and when surrounding senescent cells are firing SASP signals, it can become a tumor promoter. Campisi's 2013 review is the essential reading on this paradox. Now let's shift gears to apoptosis, which is the other major way cells can die. The word apoptosis comes from Greek. It literally means leaves falling from a tree. Beautiful, isn't it? Because that's exactly what it is, a natural, programmed, orderly process of cell death. But before we go deeper, let's compare it to the other way cells die, necrosis. Necrosis is accidental death, caused by injury, toxins, or infection. When a cell undergoes necrosis, it swells up and ruptures, spilling its contents everywhere. This debris triggers a full inflammatory response in the surrounding tissue. It's chaotic and damaging. Apoptosis is the opposite. It is completely controlled and intentional. The cell shrinks, its nucleus and chromosomes fragment, it breaks into neat little packages called apoptotic bodies, and these are quietly cleaned up by nearby immune cells called phagocytes. No inflammation, no damage to neighbors. This elegant process was beautifully described by Taylor, Cullen and Martin in their 2008 paper in Nature Reviews Molecular Cell Biology, titled Apoptosis: Controlled Demolition at the Cellular Level. A must-read paper. So why does apoptosis matter? Let me give you some incredible examples. Example 1, your fingers. When you were developing as an embryo, your hand started as a flat paddle, with webbing between the developing digits. Apoptosis destroyed that webbing to give you separate fingers. Without apoptosis, you'd have duck hands. Example 2, your brain. This one blows people's minds. The majority of neurons in your brain die during development via apoptosis. Only the neurons that make the best connections survive. It's a brutal quality control system, but it's what gives you a precisely wired brain. Example 3, your immune system. In the thymus gland, T cells that fail quality control, either because they don't work properly, or because they would attack your own body, are eliminated by apoptosis. This is called clonal selection. Without this, you'd have autoimmune disease. Example 4, cancer prevention. Every time a cell accumulates dangerous mutations, apoptosis is supposed to kick in and destroy it before it becomes cancerous. Cancer cells evolve to switch off apoptosis, making themselves immortal. Understanding how they do this is key to developing better cancer therapies. Now, how exactly does apoptosis happen? There are 3 main pathways, 2 extrinsic and 1 intrinsic. You can actually see all 3 illustrated in your Gray's Anatomy textbook as Figure 1.29. Pathway A, the granzyme B pathway, extrinsic. This involves your own killer immune cells, cytotoxic T lymphocytes. When they detect an infected or abnormal cell, they release a protein called perforin which literally punches holes in the target cell's membrane. Through these holes enters granzyme B, a protease that acts like caspase and directly triggers cell death by activating caspase 8. Pathway B, the Fas ligand pathway, Fas L, extrinsic. This is a death signal sent from outside the cell. Fas ligand binds to death receptors on the cell surface. This recruits an adapter protein called FADD, Fas-associated death domain protein. FADD then recruits and activates caspase 8, which kicks off the death cascade. Pathway C, the cytochrome C pathway, intrinsic. This one starts inside the cell, triggered by internal stress like DNA damage. BX-BK channels open up in the mitochondria, the cell's powerhouse. Cytochrome C is released from between the inner and outer mitochondrial membranes into the cytoplasm. It then forms a large protein complex called the apoptosome, which activates caspase 9, which activates caspase 3, and the demolition begins. All 3 pathways ultimately converge on the effector caspases 3, 6, and 7. Let's talk about caspases in a little more detail, because these are the actual molecular machines that carry out apoptosis. The name stands for cysteine aspartic acid-specific proteases. In plain English, they are molecular scissors that cut specific proteins inside the cell, systematically dismantling it. There are 2 types. Initiator caspases, numbers 2, 8, 9, and 10. These are present in healthy cells as zymogens, inactive precursor enzymes. Think of them as locked weapons, just waiting for the signal. When a death signal arrives through any of the 3 pathways we just discussed, these initiator caspases get activated first. Executioner caspases, numbers 3, 6, and 7. These are activated by the initiator caspases. Once active, they go to work cutting the cell's cytoskeleton, causing membrane blebbing. The cell surface starts bubbling. Nuclear fragmentation. The nucleus breaks apart. Apoptotic body formation. The cell packages itself into neat fragments. And complete cell disassembly. As Taylor and colleagues described, caspases use a death by 1,000 cuts strategy. They target hundreds of proteins simultaneously, ensuring the cell cannot survive and cannot fight back. Let me take a moment to acknowledge the brilliant scientists whose work forms the basis of today's video. Cubbon N. and Mistil IT, 2017, published in Nature Reviews Molecular Cell Biology, Volume 18, pages 595 to 609. They used progeria syndrome as a window into understanding normal aging, showing shared defects in genetics, epigenetics, and metabolism. Campisi J., 2013, published in Annual Review of Physiology, Volume 75, pages 685 to 705. This is the landmark paper on senescence as both a tumor suppressor and tumor promoter. If you read one paper from today's video, make it this one. Taylor R.C., Cullen S.P. and Martin S.J., 2008, published in Nature Reviews Molecular Cell Biology, Volume 9, pages 231 to 241. Defined apoptosis as controlled cellular demolition and explained exactly how caspases orchestrate cell death. Tuttle et al., 2021, documented how senescent cells accumulate in aged tissues and directly cause diseases like Alzheimer's, osteoporosis, and renal failure. All of these are open access or available through your institutional library. I highly recommend reading them. Let me quickly summarize the key differences between senescence and apoptosis, because students often confuse the two. Both involve a cell-stopping division, but after that, they are completely different. Senescence. The cell stays alive. It's arrested, yes, but it's still there, still secreting SASP signals, still causing inflammation. It's a slow, gradual process. Apoptosis. The cell dies completely. It's rapid, clean, anti-inflammatory. No mess, no harmful signals. Senescence is irreversible. The cell cannot be restarted. Apoptosis is also irreversible, but for a different reason. The cell is gone. Senescence causes inflammation through SASP. Apoptosis prevents inflammation. Apoptotic bodies are cleared before contents spill. And importantly, some senescent cells become resistant to apoptosis. They refuse to die even when signals tell them to. This makes them even more problematic in aging tissues and in cancer therapy. Alright, let's bring it all together with 5 key points to remember from today's video. Point 1. Aging is a gradual functional decline driven by cellular hallmarks, most importantly telomere shortening and cellular senescence. Point 2. Senescent cells are permanently arrested, still alive, and actively secreting harmful signals via SASP, contributing to age-related diseases. Point 3. Senescence is a double-edged sword in cancer. It can prevent tumor formation, but it can also promote cancer progression in neighboring cells. Point 4. Apoptosis is controlled, clean, self-destruction, essential for development, immunity, and cancer prevention. Point 5. 3 apoptosis pathways exist: granzyme B, FAS-L, and cytochrome C, all converging on executioner caspases 3, 6, and 7. If you can remember these 5 points, you have a solid grasp of one of the most clinically important topics in modern biology. And that's a wrap on today's video. If you found this helpful, please give this video a thumbs up, share it with your classmates, and subscribe to Gyaan so you never miss an episode.